Well, hello guys. So this is a quick look at the solution to the exercise I gave you. So you have to tabulate some differences between Euglena and Spirogyra. So you have Euglena on the left column, Spirogyra on the right. So let's look at some of the lines of difference between them. Euglena has flagella present or flagellum present. Spirogyra do not have. Euglena do not have spiral chloroplast, which is present in Spirogyra. Euglena can have rounded or sometimes stellate or star-shaped chloroplast. So if you can write that in place of the non-spiral chloroplast. Euglena has pellicle. Spirogyra do not have a pellicle. There's a large cytoplasm in Euglena. That of Spirogyra is reduced, so it's very thin. There's absence of a slimy layer on the Euglena. We have that in Spirogyra. That is the outermost layer. Gallette is present in Euglena. Gallette is absent in Spirogyra. Euglena is unicellular. Spirogyra is filamentous, made up of many cells, cylindrical cells that are joined end to end. So Euglena has these photosensitive receptors called the stigma or eye spots, which is present in Spirogyra. Absence of cell wall in Euglena, the presence of cell wall in Spirogyra. In Euglena, we have the nucleus not centralized. It can be found at different parts of the cytoplasm. In the Spirogyra, we have a centralized nucleus. That's due to the structure of the cytoplasm, right? So the thin cytoplasm then sort of creates a space within the centralmost area of the Spirogyra, and then the nucleus is present there. Okay. All right, so that was the first one. The second one was state the plant-like and animal-like features of Euglena. So the plant-like features of Euglena, you have chlorophyll for photosynthesis, or sometimes for practical setting, you can say green. It is green for photosynthesis. For practicals, you are dealing with the specimen itself. In as much as you cannot feel the chlorophyll or even the chloroplast. So it's not high grade for you to say chlorophyll or chloroplast. Yeah, we know they are there. The chlorophyll is there within the chloroplast. It's pigmented and all that, but you cannot feel the pigment. So it is safer in practical exams biology to say green for photosynthesis or to produce food. What have you done? Chlorophyll or chloroplast. So you can take note of that. You also have these starch storing organelles or starches. That's the paramyelon granules for storing starch. As you know, plants store excess glucose as starch. All right, what about the animal-like features of Euglena? So there is a presence of gallets for heterotrophic nutrition or phagocytosis. Then, of course, there is flagellum for locomotion. So the chief reason for a prototist to be considered animal-like is its ability to locomote, right? Any other feature is just an addition. So chiefly is the presence of the flagellum that makes it an animal. But all these other points are worth considering. Then flexible but rigid pellicle made of protein strips. These allows for movement and give shape. I don't think this is give shape, right? Give shape to, give shape, give shape that slipper, right? That foot shape or slipper shape of the sorry, that is not slipper shape. The slipper shape is paramecium. Sorry about that. I was thinking about that. Yeah, so basically the Euglena has two ends, the anterior and then posterior ends. The anterior ends are usually blunt, rounded, whilst the posterior end is larger and tapers to the end, becoming smaller and narrow. All right, so so the pellicle is to strengthen. And why is it an animal? You know that most animals, like if humans, for example, they have a skeletal system or the cartilaginous structures that tend to give it shape and strength, So that's what it also has to do the same function. And as I said, there's the eye spot or stigma. Eye spot or stigma. The eye spot or stigma that detect light to move toward or away from light sources, a behavior known as phototaxis. So this feature also gives it an attribute close to animals. Animals are highly sensitive, to stimuli. As you have said, irritability. It is more developed, enhanced in animals than in the other groups of organisms, plants, fungi, prototists, and so on, or protists. All right. And then another important feature that related to animals is the presence of the contractile vacuole. Contractile vacuole for osmoregulation to expel excess water, maintaining osmotic balance. That's what happens in the kidney of humans in order to sort of regulate the amount of water in the blood. All right, so guys, this is what I have. I'll see you in the next one.